Good morning and welcome to the ZP Developer Zone. So we like to do the ZP Developer Zone every Thursday at 8 a.m. London time, and today I know we've started 10 minutes early. We often, unfortunately, we do get busy during the week, and today we're at a conference, so we've started a little bit early. But let me go straight into it. I think I put this slide up more out of tradition. We have ZP Academy, a couple of free courses there. People find them useful. We do the webinar. If people want free advice, or we're sort of helping out in some academic programs, then we sort of use the webinar to give advice. Our advice is slightly different, I would say. It's kind of pragmatic advice. And what do I mean by sort of pragmatic advice? We don't have an academic sort of perspective or an agenda. Just want to get the job done, if I can put it that way. So we do have these webinars. We do collaborations. Collaborations, unfortunately, do have to have some sort of funding with them, so it does work in certain jurisdictions more than others. We are recruiting at the moment. We're mostly recruiting from the USN, University of Southeast Norway, because one of our sites is in Norway. Though today I'm actually calling you, or speaking rather, from Swansea, because we have a location here, a site here as well. We do have our ZP Developer Zone, and we do have workshops. The workshops are kind of super relevant to some of the questions today, because the workshops, if you come to Norway, in fact, I'll go to the next slide. If you come to some of our Norwegian workshops, by the way, every time you see a link, hopefully, this one I might have actually forgotten to do, is put underneath the video. But if you come to Norway, those are 2-day workshops. I think they're really intense. We will, I hope, enjoyable as well. The ones in UK tend to be 1 day. But we can answer technical questions there. So some of the questions that start to come and get quite deep, and at some point, somebody has to come and kind of see us, and so we can kind of go through this one-on-one. But that said, we do have our workshops. Question today: what is VoltDeposit? VoltDeposit is something that we've recently released. I'll touch upon it. Materials for biosensor manufacturing. Very quick question: what do you manufacture on? I'll answer that question. Electronics for gas sensors. We're working with some people at the moment for doing gas sensing. A lot of what we do is actually in the liquid phase. I am going to touch upon it that somebody's interested in doing electrophysiology. That's the sort of study of things like ion channels on the cells. That's very much liquid phase. So I'd say 90%, if not 95% of what we do is actually in the liquid phase, but we do some gas phase work as well. We will talk about pH sensors. pH is the most measured parameter in the world. It's interesting because this conversation that I'm having on pH sensors, which is a good group of academics, or academic based, they're really starting to, I would describe it as getting to product development, actually. So I will touch on that a little bit today. As I say, we do have a question that's in from electrophysiology. The inquirer has actually come to the same conclusion that we've also come to, that actually electrophysiology and electrochemistry, we're all using the same equipment. What's different is, and I have found articles on this, and I'll go into the history of this, but we're doing the same stuff. We're actually using different equipment, but the equipment is doing the same thing, and I will dig into that a little bit deeper today. And then somebody did ask about 510K. I will answer. It's a short question, but the 510K is a way of getting onto the market, receiving kind of, I want to say FDA approval, but I'll check if the word approval is correct. But it does allow you to get onto the market in the USA, and I will touch upon 510K. Actually, I think these list of questions is actually quite nice, because it kind of demonstrates some of this is equipment and manufacturing, some of this is definitely the science, some of this is applications, and one of these is actually regulatory. So that kind of, the whole, how do I say it, the whole thing that ZP does. I realize there's actually a question missing here. I must have 2 question 5s or something where I talk about commercialization. So I apologize if you're the person asking about kind of veterinary applications, how to develop those, then I definitely have covered that in here as well today. So VoltDeposit. VoltDeposit is, in one way, VoltDeposit is actually the fusion between robotics and potentiostats. And what I mean by that is we do have some videos. I do have a couple of links to this, but at ZP, we are ISO 13485 contract developers, contract manufacturers of electrochemical biosensors. And in order to do that, we do have robotic systems. we're also developers and manufacturers of potentiostats. And really what we've essentially done is fused our potentiostat capability with our robotic capability. So now, rather than the science or the manufacturing necessarily being quite manual, we've actually put the potentiostats essentially onto the robot. So rather than me doing this, the robot is doing this. And the robot's actually doing it at 40 electrodes at a time. One of the questions today is actually I think it's more of an electrophysiologist. So in biology, they do a lot more testing than maybe in science and maybe than chemistry and material science. So what I mean by that is they will do what's called 96 wells. They will do a lot of repetition of experiments to make sure they're statistically significant. And traditionally they'll do like 96 experiments in a well plate. And chemistry and material science doesn't tend to work that way, but actually it should. And so VoltDeposit is a way of automating discovery and, by the way, manufacturing. And as I say, it's really doing things that we would have done electrochemistry-wise, one at a time, maybe on a parallel potentiostat. But now, rather than me moving electrodes around the lab, the robot is moving electrodes around the lab. And once you start doing that, now you're starting to be able to sort of automate the synthesis or fabrication upon electrodes, and then you're able to automate the testing of electrodes. And of course, we can just send that data to the cloud. We do have Julie, and we do have AI capability in Julie. So suddenly, it's a sort of singularity that things that we were doing separately, you bring them together. So the robot's something we were doing separately, the parallel potentiostats was something we were doing separately. Julie, which is a cloud database for the collection of data, the analysis of data, sharing of data, so we do separately, bring them together, and you're like, oh, hang on, the value of these three parts is much greater when brought together. Anyway, so I've just put a couple of links there because I realized those videos weren't necessarily linked. But VoltDeposit for us is really about performing experiments in a sort of precise and consistent manner. It just makes for better data quality. And we're not just doing, and I should have said this really, that we're not just doing one sensor. We're actually loading up these, maybe I call it a printed circuit board carrier with 40 sensors. What it means is that we can actually fabricate 40 electrodes at the same time, do some electrode deposition, electropolymerization, electroplating. We got all of that data. Once we've got data and we start getting data on scale, then you can actually start using AI, because now you can sort of produce enough data to start training the AI. And something that we have anyway is Julie. You don't want to crunch this. You don't want to be crunching this kind of data necessarily on the local PC. When you're doing this kind of stuff on a PC, it's isolated, it's locked on that computer. Now send it to the clouds, because when you've got the cloud, you can have logins. We've even brought in the idea that people can review data. So it does have cloud capabilities. And as we say, we have something called Julie. I haven't linked to Julie today, so unfortunately that's something you might have to Google if you're kind of curious about that. Now I think I'm getting a bit, this is very kind of, that was a very broad concept that I was just describing there. This is very specific. The gentleman's asked, what substrates, you're a manufacturer of biosensors, which materials are used as a substrate? If I could just slightly change the question. So the quick answer is, use one of our screen printed electrodes. We use a whole range of materials, from polymers to PCBs, paper in our time, and historically the oldest material that we've used is actually a ceramic. Here's the data sheet. I know I'm slightly obscuring it, but don't worry, because I've put a link here as well. You can have a look at that. So obviously we have to be careful about these kind of questions, because it's a commercial organization. ZP, we try to do a lot of good by putting a lot of material into the public domain, but we are a commercial organization. I think this is being transparent, but if you want to have a look at the data sheet is there. Question number 3 is really about electronics for gas sensors. It's really just that we do have a gas sensor. For example, this is carbon monoxide, and we do a lot of gas sensing. This is the test jig. It's always fun working with gases like carbon monoxide, because they are quite toxic. Just want to make sure that everything is, yeah, just making sure that everything is going to the internet. Okay, so we do these test jigs. We do handle, unfortunately, toxic gases like carbon monoxide. The only reason I'm bringing this up is because we were historically using some electronics. We made these commercially available, but we are actually doing a transition now over to some new electronics. So if you're a sort of developer, especially if you've got more of an engineering or electronics or electromechanical background, then this would be the kind of electronics that we would be asking you to use. Question number 4 is really, this is probably the longest question I have today. And what's really good about this, and I want to say a personal thank you, that the team that sends me this stuff, they present it in a really clear presentation. You know, so they're doing the work for me and telling me their story. So, you know, somebody sends you, you know, it doesn't work email. That's really tough to deal with. Or somebody just sends you some data, no axes on it, no context. You know, it's really hard. But these people, to be absolutely fair, they are describing their setup. I support these people on their setup because, you know, they've been working with us for a little time now, but they are not using our electronics, which makes it tougher. But I just want to sort of full transparency, they're not using our electronics and they're not using our connectors, but they are using our sensors. The reason I kind of sort of bring that up is because if you're now watching this, then I won't, and you're a new customer, I would be reluctant to support it because now I'm having to troubleshoot and I'm not sure if it's the electronics. In this case, I believe these electronics, their setup's very clean, so I don't have an issue with it, but I'm just saying that I won't be so super keen to support their electronics, especially as we do have our own equivalent to everything that was actually in that image as well. But of course, I will support people who have been with us and working with us for a little while. First thing I want to do is sort of set some expectations. So this is some pH data of ours. So what's going on here is I have potential measured using a parameter called open circuit potential, and this is time. This is at pH 7, this is at pH 9, this is at pH 4, this is at pH 7. So I'm quite happy with the way that the pH is changing. Now if I focus in on that pH 7, if I focus on these two different data sets taken very different times, but it just sort of tells me that there is a consistency and a repeatability and a reproducibility. So all these kind of words in my head, no, we can kind of carry on doing this. So let's have a look at this pH 7. There's about 5 sensors in there, and the spread in the data, the max and min, is 25 millivolts. If I look at another set of data, the max and min there is 50 millivolts. So it tells you that sensors have a variability in them. I often find that pH sensors, their change to pH, if I can call that their sensitivity or the slope, is quite conserved. It's actually the offset that is variable. So I have a sort of expectation of variability in my head. So I want to help these people. Excellent presentation, really clear, you know, and this is their work. And I want to just sort of say, you know, I can help you most when people do this kind of work. So thank you so much. Now, what's interesting to see is if I look at that pH 7 data, there's definitely the first one they ever do is like that. It's kind of crazy. But then the rest of their pH 7 data is all down here. So it's kind of 75 millivolts of spread earlier on, which is not bad because I was saying on my previous slide, I would expect a spread of 25 to 50. That first one is kind of crazy. And then they have actually a whole bunch of repeats that are at much less, well, actually about 20 millivolts spread down here. Their pH 4 is up here, which I would have actually expected. So I kind of get this data and it makes some sense to me. And then they have a very, you know, clear, a similar story again, where they have repeats at pH 7. And then their pH 4 is up there. Their pH 7 has a spread of 70 millivolts. But in fact, if I ignore the first reading, subsequent readings are actually much closer. In helping these people, we've got a bit of a correspondence going, and I've said, all right, go into the lab and do it exactly the way that Andre does it in this video. Also, Andre is kind of talking about the instruments he's using. He has these solutions and he's moving the sensor from solution to solution. So I've kind of described to them, do an experiment like this. And to be fair, they did it, you know, so, and I think they maybe even did it this week. But I've seen it, or maybe it's just the end of last week, they did it. But now they did have some curious results, and I'll slightly drag myself out of these curious results. The pH 7 is fine. The pH 4 changes. By the way, I think you've actually swapped the pins on your leads, because earlier on you were showing that pH 4 was the highest, and then pH 7 was the next highest. Now that's actually changed. I think your pins might have slightly changed on the way you connected, but if you say no, then I believe you. But anyway, pH 7, pH 4, so it changes, and then disappointingly, the pH 9 is not in the right place, and that is a problem. So that's something I need to address. So, you know, I recognize. So they were doing some data. I looked at it. I thought, oh, this is not looking too bad. I tell you what, I'll get them to do. I'll get them to do exactly the same experiment as Andre. They did it, and they did. You know, nicely labeled it up. I understand what they're doing. So look how well annotated it is. Really, really good. So I'm thinking, right, what's going on here? So start to think about the reference electrode now. So the reference electrode, so we have a pH sensitive working electrode and we need a stable reference electrode. Now that reference electrode, its potential is versus chloride. And I asked them about their buffers or their test solutions, and they sent me the data sheet and a comment. So they've obviously read the data sheet. There's no chloride in there. So obviously, at ZP, we are ISO 13485 contract developers, contract manufacturers of electrochemical biosensors. We test things like blood. Now, when you're testing blood, the chloride is stable. So if I want to measure the pH of blood, I can be pretty sure that the chloride is actually quite stable in that particular example. I mean, another one that is kind of quite easy to say on is the pH of seawater. You know, the chloride is high, but it's quite stable. But if you don't have any chloride in your sample, which I don't think they do, the reference electrode essentially doesn't know what to do. Now let me go into that a little bit deeper. That I would describe as this whole discussion would be 20 to 30 minutes worth of slides, but I have to summarize it a bit. So you want a pH potential that is stable, is not variable with the sample, but silver chloride reference electrodes, which are used a lot in screen printed electrode formats, actually have the reference electrode if you have very little chloride, the potential is one thing. If you increase the chloride, the potential is another thing. These people essentially have no chloride, so it's probably very crazy what the reference electrode is actually trying to do. Again, if you see a link, then I will have already pasted it underneath the video, and I hope that is super useful to you. Now what's happening is you're measuring pH versus the reference, but of course if the reference is changing, then your pH, let's say that my reference did this, the pH value would do this. If my reference did this, the pH value would do this. So some of your oddness is not coming from your working electrode, it's probably coming from your reference electrode. How do we fix it? Okay, this is something that we do at ZP. It's not perfect, but I will describe it. I think I'm going to give you 4 ideas. One of them is you take a pipette and you functionalize the reference electrode with something that we call the chloride resilient formulation. The chloride resilient formulation is essentially chloride anions bound up in a polymer. So we're trying to turn what's essentially called a pseudo reference electrode into a true reference electrode by fixing the chloride concentration. So that is a way. I'm slightly reluctant because it takes some effort from you, and it will give you a more stable reference electrode, but eventually the chloride will diffuse out of that reference electrode. So if you're looking for sort of, I'll be extreme, 2 weeks of continuous pH operation, it's not going to work. Unless the sample has a true fixed chloride concentration. So let me just correct myself. Say, if you were trying to do a 2-week measurement in seawater, all my problems go away because the chloride is pretty fixed in seawater anyway. But idea number one is to put a polymer onto the reference electrode that has a chloride reservoir bound up in that reference in that material rather, and then the reference goes from being a pseudo reference to a true reference electrode. Number two, right? This is, as I did mention that we're really starting to get into product development now. This is the tricks and tips that CP does, you know. So somebody has a sample, maybe it's urine. I want to measure the pH of urine, but actually urine has a variable chloride concentration. So what do? I put a salt drop and I leave it to dry. So I've got a salt patch on my electrode. So then I bring in my sample of urine, which may have a variable chloride, but in fact, the drop comes in, it dissolves the salt, and I've made it so that actually the chloride that comes in with the sample is insignificant compared to my salt patch. So in fact, I do end up dominating the chloride in that sample. So I'm now controlling the sample. So you can, maybe I call this idea number two almost sample prep. Idea number one was almost was sensor preparation. Now I'm talking about sample preparation. So the salt deposit dissolves into the sample. After about a minute, it should be fairly homogeneous, and then you've got now you have a fixed way, more controlled chlorides, and you should get a more controlled pH measurement. So that's idea number two. These are product development ideas, by the way, straight from industry. Sample number three. I meant to put some references into Food Sense here, but I got Food Sense here. There's a little link here. Have a little look at this, because in Food Sense, what you'll see that we do is we take a solution that we know, and we add in a unknown solution. So we do a bit of sample prep. We homogenize these two things, and then we test it. So watch our Food Sense videos, because what we're doing there is, the question that came from this academic group was, how do I test unknown samples? Well, in our world of chilies, I'm not testing pH, I'm testing capsaicin, but I've got what's called a matrix effect. Every flipping chili source in the world is variable, so how do I deal with it? I actually take a small sample of the sample, let's say, and I put it into a liquid that I control. So I dominate the matrix. So sample number three is, again, sample prep, but it's, I would say, watch those videos on Food Sense to get a sense of what we're doing on Food Sense. Idea number four, I think this is probably one of the better ones, because I realized that when you take, if you're trying to measure pH, you take a sample of unknown pH and you put it in another solution. I'm worried about some sort of strange buffering between these two solutions or changing the pH. So I think another idea actually is to have a container, have a salt, how would I put it? Have a mass of salt in there that you know about, and then just put the sample into that. So you're not trying to add salt through a liquid, you're just adding salt, basically, as the crystalline form, and then you're going to have less problems with pH. Let those two things homogenize, and then you can record it. So, a couple of things. Excellent work. Love the way you're annotating the data and helping me. Love the fact that you're looking for solutions and not just complaining about problems. And then I've tried to give you 4 ideas about how to deal with unknown samples. Question number 5. This is a really interesting one for me. It's about electrophysiology. So I first really became aware of electrophysiology about 15 years ago. I was in San Diego. I was at an expo. I was talking to the guy next to me, and he started describing ion channels and how they would open and close ion channels in the cellular walls, and they would get these electrical signals. And I just listened to this guy, and I thought, I tell you what, he's describing electrochemistry. And there I was with my PhD in electrochemistry, but I'd never heard of this entire field. Fast forward now 15 years, and I realize, and I have found articles on this, that there are people doing electrophysiology and there are people doing electrochemistry. We're actually doing different samples, but we're doing actually the same experiments. Now, this person has sent me a paper and sort of asking the questions, can I use your technology to do this paper? The first thing about this paper is they do use a potentiostat from CH Instruments, so it immediately tells me, yeah, they're using, rather than technology that's been marketed as electrophysiology, they're actually using technology that's been marketed towards electrochemistry. And because it's CH Instruments, of course I would know, I'm supposed to know my electrochemistry and my electrochemistry vendor, so I do recognize it. I actually own CH Instruments because we bought a company that went into liquidation. We bought the assets and we got a couple of CH Instruments. I don't recommend CH Instruments in this case because I am biased. I manufacture potentiostats. So, you have to make a declaration of bias, and that's my bias. But the fact that they are using CH Instruments does tell me a lot about. They are essentially using a potentiostat or an EIS, potentiostat electrochemical impedance spectroscopy, to do this work. The problems I have with this paper, apart from their use of their brand of potentiostat, and that's because I'm biased, is the way they make this gold electrode. They make this gold electrode. So rather than the focus of this paper being on the, I would say on the biology and electrophysiology, they go and make this quite complicated electrode. They sputter it, and then they form a self-assembled monolayer on this gold electrode. And they use things like chloroform, which is fine, but the value add in this kind of work is actually the electrophysiology. Making a gold electrode is not value add. So we have gold electrodes. I will link to them in a bit. Now, to re-emphasize my point, that electrophysiology and electrochemistry, I don't want to say it's the same thing, but we are using the same instruments. Electrophysiology, I've got to kind of quote it from a paper here, counter reference electrode, and we have a very similar, if not the same, if we have the same setups also in electrochemistry as well. And having read that paper, yes, they are using a EIS, an impedance spectrometer, and they're gathering Bodier, and they're using equivalent circuits. It's just the person who asked this question, myself, and the authors of this paper have all realized that if you own a potentiostat, then you can do cell electrophysiology work as well. So his questions were quite specific. Can you use the Anapot EIS instrument? Yes, you can. He's asking, should I use the hypervalue carbon electrode? Having read the paper, they weren't using carbon, they were using gold, and they had a formulation for putting self-assembled monolayers onto that gold. I don't like the science of what they did because some of those solvents are quite aggressive. Now we do self-assembled monolayers, but we never use these kind of organic solvents. We're using much more buffer aqueous-based systems, so that I have a problem with, and I have a problem that they're functional. They fabricated the gold electrode. It doesn't add a lot of value to fabricate gold electrodes. If you see a link pop up, then I've hopefully already put the link underneath the video. And then he's asking, can I hold this potential and do an EIS experiment? Yes, you can. We do this all the time. In fact, come to one of our workshops because we do this kind of experiment at the workshop where we kind of hold the potential and then we do EIS. By the way, it's very boring, very dull statement. We call it Faradaic impedance spectroscopy when we do that. I think I had ended up not listing this question, but somebody was interested in developing point of care. So Zimmer and Peacock, I think one of our big raison d'etres is to turn lab research into products. So somebody develops a sensor on a gold electrode. This particular questioner was actually more interested in veterinary applications. I'm super interested in veterinary applications. I love veterinary applications. They have a very different route to market. Obviously, human health is very regulated and veterinary health is less regulated. So what's the workflow for getting R&D experiments into products? At least if you're working with ZP, we have the kind of gold electrodes. We think we make really good gold electrodes. For example, if you're trying to make an immunosensor, you probably often use a gold electrode, and we can get signal versus concentration, and we can get some really nice repeatability. I do have these slides when we do our workshops, but I'm just going to sort of describe here that when we take electrodes and we're trying to make either attach cells to them or trying to attach antibodies to them, we have gone into this a lot, that we end up functionalizing the electrodes so that then when things like the antigens bind to the surface, we can either pick up that signal using impedance spectroscopy or other techniques like differential pulse voltammetry, square wave voltammetry. I should say that the instrument I was talking about, Anapot EIS, earlier on, actually will do essentially all of these techniques. I'm just bringing this to say that what ZP does is, somebody sometimes comes to us and says, oh, I want to make a veterinary test. Great. Is it molecular? We can deal with that. Is it small molecule? We can deal with that. Is it proteins, hormones? We can deal with those. So we have lots of ways of detecting molecules that are in, or biomarkers that are indicators of diseases. Then we want to start getting into a product. So we would take like our electrodes, maybe we'd put them into an array. So here we have something that's a credit card size. Hopefully I have put links for this, and then these are all the sensors here. The sample goes in, and then we can actually do multiplex testing on that sample. And we even have instruments that can take that credit card. And yes, I did put some links to that. So I suppose when we first started in ZP, we had this idea that we were going to, and you do find it sort of still in our website, of taking ideas to market. I think I even say it in a video that, there's plenty of great ideas in academia, but unfortunately, few of them actually make it to market. All right, let's set up a business that can be part of that journey. Question number 6 is, somebody's question was quite straightforward, which is, are your microneedles 510K approved? All right, so we do have microneedles. Here's a link to them. Hello, I would like to know if your microneedle arrays are cleared by the FDA with a 510K. So a 510K is one route to market for a medical device or an in vitro or a diagnostic. The way the 510K works is, the easiest example I can give you is a glucose strip. So there are plenty of glucose strips on the market. So, and they're FDA approved. So what you do is you say, right, I'm going to buy these glucose strips that are already approved, and I'm going to test them. And I'm now going to test my glucose strips that I would like to approve. And I'm going to demonstrate to you that the data I get from the approved glucose strips, I get the same data on my glucose strips that are not approved. And then you write a report for the FDA and you say, look, these approved ones work like this. My ones work the same way. Will you give me a 510K? And they will say, if you do a good enough job and your reporting is good and you answer their questions correctly, they go, stamp cleared. So a 510K works when there's what is called a predicative device on the market, something that's already in the market, is known to work, it's quite robust, and you show that you're equivalent to it, and then you get a 510K. So the question, unfortunately, is slightly wrong, because we've got microneedles, but microneedles to be FDA approved you have to then specify what the application is. There are very few microneedles that I would say are FDA approved. I know about the healthcare industry where they use sort of versions of microneedles and needles to do Botox, for example. But the question whether our microneedles are 510K approved, the answer is no, because they have to, when you ask the question, you have to say 510K approved for this application or that application, then you go and look for that predicative device. But what I can say about our microneedles is we are ISO 13485, and we help companies functionalize microneedles in order to use them in medical applications. And as those customers come through the pipeline, 510K may be a route for them. But because there are very few sort of microneedle type approved products on the market, I suspect that 510K is actually not a route for those customers that we have. So question number 1, what is VoltDeposit? VoltDeposit is this kind of amalgamation between robotics and potentiostats. And I'm sorry that a few people have come online and found out that we started early. I say we haven't had to do this because the days are pretty busy and I'm at a conference today. Materials for biosensors, I've definitely given you the link. Have a look. You can read that data sheets, enjoy yourself. Electronics for gas sensors. We are shifting from an old set of electronics to a new set of electronics. pH sensors. Their question really is, how do I measure an unknown sample? Now knowing that chloride is part of my signal, I need to fix that chloride. And I've given you, I think, 4 pretty good ideas and a link to food sense, just to give you a sense of what we're doing, let's say, in the real world, when you can't. I love laboratories. You can always control the sample. You step into the real world, bang, you cannot control that sample. Or you have to, you do have to take means to control it. Electrophysiology, I read that paper. Yeah, they're definitely using any impedance spectrometer. Yeah, we have our equivalent. 510K, useful question, because it does demonstrate, even in this webinar that actually we could talk about every aspect along the idea of from idea and prototyping, actually even to FDA approval. So the quick answer is it's not 510K approved, but I have given you quite a detailed explanation that you have to look for an equivalent product. And on microneedles, there's not so many equivalent products out there already. And then I've got to contact us. So I would just check. And I want to say thank you very much. My apologies for starting early today. I've said it, several times that we're at a conference today, and therefore we have to kind of get moving and get to that conference. But thank you so much for the questions. They are appreciated. And if you have any questions, essentially don't hesitate to reach out to us at ZP. Okay, thanks very much.